This is Matt Dean with A+ College Ready, and today we're going to talk about plasmids and restriction mapping. Plasmids are circular pieces of DNA found in bacteria. Bacteria can exchange plasmids during conjugation. That's where they hook together and exchange genetic information. This process is really important because it gives them a source of genetic variation, because typically bacteria reproduce asexually through binary fission. Scientists also discovered a few years ago some special enzymes in bacteria. They're called restriction enzymes. These are enzymes that cut DNA at very specific sites known as restriction sites. Now you might be wondering, why would bacteria have enzymes that cut DNA? A lot of scientists think that they're in there to help protect the bacteria from essentially invasion by bacteriophages, bacteria infecting viruses. Restriction enzymes usually have strange names like EcoR1 and Hind3. They're typically named after the bacteria they were discovered in. Here's a really famous plasmid over here. This is the P-glow plasmid. This plasmid has been genetically engineered with a gene for a protein called GFP, green fluorescent protein. But what I want to point out mainly in this plasmid right now are these little red marks. These are what we call restriction sites. So at each one of these, if for example, if P-glow was treated with the enzyme EcoR1, the plasmid would be cut at this restriction site. If it was cut with the enzyme Hind3, it would be cut here and here, and so on. Some other important things I'll point out while we're looking at the P-glow plasmid. It's about 5,400 base pairs long. It's base pairs of ATGC, nitrogenous bases. It's also been genetically engineered with an ampicillin resistance gene, an antibiotic resistance gene. It's got an origin of replication where the copying of the DNA starts. And we may talk about this section a little bit later on. So next we want to apply these ideas of plasmids and restriction enzymes to this process of restriction mapping. So here it says, by analyzing the size of DNA fragments that result after digesting, and digesting here just means essentially cutting up DNA with restriction enzymes, we can do a map. Essentially, the map shows you where the enzymes cut at and the size of fragments you would expect to get if a particular plasmid was treated with a certain combination of restriction enzymes. So the first question below is a restriction map for the plasmid PGen 101. Its total length is 20 kilobase pairs, or 20,000 base pairs. Using the map as a guide, give the number of restriction fragments along with their associated links that would result from digesting the plasmid with the restriction enzyme EcoR1, with BAM-H1, and also with EcoR1 and BAM-H1 at the same time. So if we look at the plasmid, EcoR1 only cuts the plasmid in one location, right here. So if we ran this through an electrophoresis chamber, we would still get just one fragment of DNA. It would just be cut open. That fragment of DNA would still be the whole length of DNA, which was 20,000 kilobase pairs. So we would expect to get one fragment, and that fragment would be about 20 kilobase pairs. Now, on the other hand, if we treated it with BAM-H1, that enzyme cuts at this restriction site and this site and this site. So it cuts in three places. So we would get a fragment, this fragment, that is 6 kilobase pairs. We would get another fragment that would be this whole section right here, because remember, we're not using EcoR1 right now. So we would get another fragment that's 12 kilobase pairs. And then finally, we would get this fragment, which is 2 kilobase pairs. So we would expect on this part to get 3 fragments. I'm just going to abbreviate that, 3 frags. The last one should say EcoR1 plus BAM-H1. So now if we use both, the DNA is going to be cut at all these restriction sites. It's going to get cut here, it's going to get cut here, here, and here. So we're going to get 1, 2, 3, 4 fragments. And those fragments are going to be of lengths 6 kilobase pairs, 8 kilobase pairs, 4 kilobase pairs, and 2 kilobase pairs. So that's using a map to determine how many fragments you would expect to get and how long those fragments would be. Now we're going to look at it sort of from the other direction. So here we have 2 freshman college students interested in becoming gene jocks, whatever that may be. They're performing the following set of restriction digests on a new plasmid PBLA230. And what they got when they treated the DNA with only HPA1, they just got 1 fragment. When they treated it with HIND3, they got 4 fragments. When they treated it with both, they got 5 fragments. They got 2 6 kilobase pair fragments. So let's start with a circle because we know plasmids are circles. The whole length of this fragment must be 26 kilobase pairs. And that's because when you treat with only HPA1, you only get 1 fragment. So that enzyme essentially just cuts the circle open, straightens it out. So let's start with HIND3. HIND3 is going to cut this piece of DNA in 4 places. We're going to get 4 fragments. So you have to kind of do this by trial and error and just kind of figure out what works. It's sort of like working a puzzle. So let's say that it cuts here. And I'm just going to label that one HI for HIND3. And then let's say it cuts about right here again, HI. Now I did that to be in the middle because we know the whole thing is 26 kilobase pairs. We know one of the fragments formed with HIND3 is about 13. So like so. And then we get another fragment that's about 6. So let's put that about right here. We get another one that's 4. And then the final piece is going to be 3 kilobase pairs. So that might be the restriction map simply for HIND3, treating the plasmid with HIND3. But the last one here, we're going to treat it with both HPA1 and HIND3. And notice that we still get a 6 kilobase pair. We still get a 4 and a 3. But instead of getting a 13 kilobase pair fragment, we get a 7 and a 6. So what I'm hoping that tells you is that HPA1 cuts somewhere right in here. And so this fragment's not going to be 13 anymore. We're going to get 1 fragment that is 7 kilobase pairs and 1 that is 6 kilobase pairs. So that is using the results from a restriction digestion to help to map out a plasmid, to show the restriction sites and to show the size fragments you would expect to get when you use a combination of restriction enzymes to digest a particular fragment. So let's look at another similar kind of example. Here's another example. This time we have a bacterial plasmid. It's 100 kilobase pairs in length. The plasmid was digested with 2 restriction enzymes, EcoR1 and HEY3. And then it was also double digested with both of those enzymes at the same time. We've separated the fragments using a gel electrophoresis, and we got the gel pattern that we see below. So when the plasmid is digested with just EcoR1, we get 2 fragments. 1 at 70 kilobase pairs in length and 1 at 30. When we use just HI3, we get a 60 fragment and a 40 kilobase pair fragment. And then when we use both at the same time, we get 4 fragments. So what we want to do is to use those patterns, and we want to come up with a restriction map. Again, some of this is sort of trial and error. You have to sort of figure out how the pattern works. So here's what I'm going to do first. I could start with either enzyme. I'm going to start with the HEY3 enzyme, and I'm going to say that it cuts right here and about right here. And I'm going to label those just with H's, or actually we probably ought to go ahead and put HEY3. Now I put those marks where I did because I know when only that enzyme is used, we're going to get 2 fragments. One of them is going to be 60. We'll label this one as 60 kilobase pairs. And one is going to be 40. All right, so then if I use EcoR1 as well at the same time, I know by itself I should have gotten a 30 fragment, and also I should have gotten a 70 fragment. So let's say that EcoR1 cut maybe, let's say it cut maybe right here. And then let's say it cut again, maybe over here. We know it cuts in two places because we know that with the only EcoR1, we only get two fragments. So remember, we need from EcoR1, we need a 70 fragment, and we also need a 30 fragment. So let's say that between EcoR1 cut right here and the EcoR1 cut right here, let's say that this piece is now 30 instead of 60 because we cut that 60 piece up. All right. That means this whole piece here is 70. Well, we know between here and here is 40. This whole piece was 60. So let's say that this right here is now 10. And let's say that this piece right here is 20. And let's see if that adds up to be 100. So 20 plus 30, 50, plus 10, 60, plus 40. So that's 100. So you've got to kind of take the fragments that you get from each individual enzyme, figure out where those cuts might be made, where those restriction sites are, and then sort of put those together so that you end up with a set of fragments that look like this. So we should have 4 fragments when we use both enzymes. The fragments should be length 40 kilobases, one of 30, one of 20, and one of 10. So that's what our restriction map should look like for this particular example problem. So let's do one more example. So here we've got, this time it's not a plasmid, this time it's just a long stretch of DNA. 4,900 base pairs. The arrows indicate the reaction sites or the restriction sites for 2 enzymes, enzyme X and enzyme Y. A, explain how the principles of gel electrophoresis allow for the separation of DNA fragments. So here you would explain that when you're doing a gel electrophoresis, that you first cut up the DNA, you've digested it with restriction enzymes, which cuts it into fragments of different sizes. You then load those fragments into a gel. You put it in your electrophoresis chamber with a buffer, a conducting solution. You always put the DNA on the negative end of the electrophoresis machine. You do that because DNA is negatively charged, and the negative end of that machine will repel the DNA, and it will push those fragments through the gel. Since the fragments are of different sizes, they'll be able to move through the gel at different speeds, and they're going to separate into bands based mostly on size. Charge also matters somewhat. So that's sort of how you should have approached A. B says, describe the results you would expect from the electrophoretic separation of fragments from the following treatments. Assume that the digestions occurred under appropriate conditions and went to completion. So the first one is the DNA is digested with only enzyme X. So if we use only enzyme X, the DNA is going to get cut right here. It's going to get cut right here, and it's going to get cut again right here. So we would expect to get this fragment, which is a 400 base pair fragment. We would expect to get this fragment in here, which is 1200 plus 500, so 1700 base pairs. We would expect to get this fragment, a 1300, and this fragment, a 1500 base pair fragment. So 4 fragments of lengths 400, 1700, 1300, and 1500. Now, next it asks us if we digested it with only enzyme Y. So let's clean up our mess here. If we digested with only Y, Y cuts right here. So we would only expect to get 2 fragments. This big fragment and this small fragment. Well, the small fragment is 400 plus 500. It's 900 base pairs. Remember that our whole fragment was 4,900. So if this is 900, all of these are going to add together to be 4,000 base pairs. Final part to part B, what happens if we use both enzymes at the same time? So if we use both, DNA is going to get here, 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 and here. So we're going to get a 1500 fragment a 1300 fragment, a 1200 fragment, a 500 fragment, and a 400 fragment. So we'd expect to get 5 fragments of those links. Now, I missed a piece. Part 4 says the undigested DNA. So if we just took this DNA and we put it in an electrophoresis machine without cutting it at all, we would get 1 fragment. And that 1 fragment would be 4,900 base pairs long. Last part of the question, explain both of the following: the mechanism of action of restriction enzymes. What they're looking for there is that restriction enzymes cut DNA after very specific base pair sequences, combinations of A's and T's and G's and C's. It's also important to note that sometimes they'll cut one strand and then they'll go further up and cut the other one. So that oftentimes you're left with some unpaired bases. Sometimes we call those sticky ends. And those turn out to be real important when we start dealing with genetic engineering and making recombinant DNA. The second part of the question, the different results you would expect if a mutation occurred at the recognition site for enzyme Y. Well, a mutation is a change in the base pair sequence. And we said a second ago that the restriction site is determined by the base pair sequence. So if a mutation happens, the sequence at that restriction site is going to be different, and therefore the enzyme won't cut there. So our answer here is because of the mutation, enzyme Y probably would not cut at all in that location. All right, that's it. I hope that helps.